We're going to be using 3D Molecular Design Synapse Construction Kit to look at the G protein and the neurotransmitter dopamine. If you don't remember that dopamine is it's an excitatory neurotransmitter. However, its effects on the actual postsynaptic neuron is actually a little bit different than some of the other neurotransmitters, such as acetylcholine or GABA. Again, just to be a few reminders, we have sodium in blue, sodium channels being orange, potassium channels being green, potassium itself being purple. Calcium channels, calcium being white. Snare proteins, the vesicle in the middle, this is dopamine. Protein connected to it. This is a sodium-potassium ATP pump. Okay, this is what we call it. This is a dopamine receptor, but is also a G protein. It's a little bit different makeup. You have the main body, which is the extracellular portion, and two internal ports, or three. This is the alpha subunit, the beta subunit, and the gamma subunit is that green portion. Explain a little bit more about what happens in a minute. Rest is pretty much the same. Synaptic clef in the middle. Again, reminders that if an action potential comes down the presynaptic neuron. If you need more information on action potentials, please see a different video. As the action potential reaches the end of the synaptic terminal, sodium floods in, causing a depolarization event. That depolarization event will open the calcium channels as well as the potassium channels. This will begin its depolarization, but the calcium is what we're interested in, binds to the protein attached to the vesicle that contains dopamine. That then causes a cascade of events that causes the snare proteins to attach together and the vesicle to exocytose dopamine into the synaptic clef. Okay. When dopamine enters the synaptic clef, it binds to this G protein. Instead of that causing an increase in sodium or potassium or chlorine or whatever it increases, this actually causes a cascade of biochemical reactions. It causes the release of the alpha and beta units. The alpha unit is the one we're interested in at the moment, because the end cascade of this entire biochemical reaction of that alpha unit ultimately causes the sodium channels to open and sodium to flood in, reaching threshold and causing a depolarization and the signal to continue. In a similar manner, dopamine is usually uptaken through various different through the protein channels, can be uptaken that way. There are a couple of enzymes that will actually degrade it in the synaptic cleft as well, ultimately leading it to finish. Because of its slightly unique nature, dopamine effects can last longer, which is part of why it's considered our pleasure neurotransmitter in a lot of cases.